Very close.
Well, thanks everybody for coming.
Thank you.
Thanks everybody for coming today to this joint CPB botany seminar in which we have Jenny McElwain visiting to give a talk all the way from Ireland, and we actually have a special guest introducing our special guest today. Cade has come from Boston to be at this talk. He was the nominee of Jenny to come, who was nominated by the CPB student organization and collective of CPB students in 2018, I think. So there are, I suspect, many of that cohort left. Has come back for it, so I'll hand it over to Kay to do the official introduction.
Thank you. Yeah. So I met Jenny in 2017 when I went as an undergrad to study abroad in Ireland, and I worked in her lab for a semester, and it was sort of what led me into switching fields from sort of mycology into plant physiology, things like that. So Jenny has had a fantastic career as a paleobotanist. She was a curator of paleobotany at the Field Museum in Chicago. And then she became a faculty at the University College Dublin, which is where I met her. And then in 2017, she became a fellow, or she joined the Irish Royal Society and became the chair of botany at Trinity College Dublin, which is Ireland's oldest university. And she also wrote a fantastic book on plant evolution, co-wrote the book on plant evolution, which is fantastic if anyone gets a chance to read it. And today she's here to talk to us about more paleobotany and how plants have changed and helped shape Earth and how Earth has shaped them throughout millions and millions of years. So thank you so much for coming.
So, can everybody hear me okay without my phone? I'm a bit blocked up with the cold. But yeah, first of all, thank you very much to Cade and Scott, and also Morgan for the invitation to come and speak. And also to all of you for hosting me. I've had a great visit today. I've learned loads of things chatting with everybody. You've all tired me out though, so I don't know whether I'll be able to give my seminar. So today I'd like to talk about, I suppose, my twin passions, using fossil plants to reconstruct atmospheric evolution, and then using the plant fossil record to try and understand how the evolution of atmospheric composition has influenced plant evolution. So I'm very much going to speak today about plant-atmosphere interaction, with some comments on, I suppose, the relevance I see with paleobotany for where we are today in a globally warming world and into the future. But mostly I'm going to just focus on the deep past and understanding the geological past. So I'm really interested in how we can use fossils from a functional perspective. How do we bring dead things to life? All we have in the fossil record is usually, we can observe anatomy, we can observe morphology, we can measure carbon isotopes, because a lot of the fossils I work with are called compression fossils. So here's a beautiful relative of ginkgo on your left. And it's a really dissected, elongated form. And I work with paleoartists to try and reconstruct what the habit of those plants were in the past. But what I really want to speak to you about is how we can use functional traits. So use contemporary ecological methods to add a functional understanding to these beautiful fossils that we can see in the fossil record. So in the first part of the talk today, I wanted to give you an overview of how we use fossils to reconstruct atmospheric composition. And I don't usually do this, but I'm going to step back in time. I usually start in the oldest and come forward, but I'm going to step back in 100 million year increments. So take you on a bit of a geological field trip. And we'll look at how CO2 changed through Earth history and how we've reconstructed carbon dioxide using fossils. And then in the second part of the talk, it's newer work, where I want to give you a few examples from case studies of how we're using a functional trace approach in paleobotany, basically to bring these fossils to life. So most of the work to reconstruct atmospheric CO2 relies on what we call proxy work. So I suppose I need to define what is a proxy, and specifically what is a CO2 proxy. So it's any biological or geochemical property of a fossil or mineral that responds to the concentration of atmospheric CO2 when they're formed. And there are loads of different proxies. There's oceanic proxies, there's paleosoil proxies, but obviously my focus is on plant-based proxies. And it makes sense. Plants are, so they're sensors of their environment. I know people in this audience work on things like carbonic anhydrase, the sensor for CO2. So it makes sense from first principles that plants should be a good proxy for CO2. So the way, basically I look at a relationship between CO2 and total conductance of the leaf surface. Usually looking at the density of stomata on the leaf surface. And I use a very much collection-based approach. So I use herbarium data sets. These are wonderful repositories of hundreds of years of dried plant specimens that can be tracked across the anthropogenic rise in CO2. So I use herbarium data sets to calibrate the CO2 proxy that I use on fossil plants. I also use an experimental approach where we expose living plants and living plant analogues, which have a deep phylogenetic history, to different atmospheric compositions. I've worked with elevated oxygen, subambient oxygen, elevated CO2, sulfur dioxide. But mainly today, I'm going to be speaking about carbon dioxide. We've also looked, to develop the proxy, we've also had to look at how other atmospheric gases perhaps influence the fidelity of the proxy. So oxygen also varied through Earth history, but it changed on a much slower temporal scale. So we've done a lot of experiments with elevated and subambient oxygen, and this gives you an idea of what our Mesozoic experiments look like. So these are plants which would have been around at the time of the dinosaurs in the Triassic and Jurassic 200 million years ago. And Harry there, one of my postdocs, is in a breathing hood because the oxygen levels we were working with would literally kill you. So it was a terrible risk assessment we had to do for that, but we use breathing hood when we're working in the plank growth chamber. So the proxy, the CO2 proxy is basically based on stomatal conductance, how plants control CO2 diffusion into the leaf and water loss from the leaf surface. And to give you an idea of one of the training data sets that we use, a lot of work has been done on ginkgo. Ginkgo has a really long fossil record. It was really speciose in the geological past. I see your ginkgos are coming into leaf out on campus, but we've got beautiful ginkgos. There's a ginkgo we collected in Yorkshire Jurassic, about 178 million years old. And you can see there that there's a relationship between stomatal index, the number of stomata ratio to the number of epidermal cells, and CO2. And this is from both a herbarium data set and experimental data sets. And then A is plants grown in ambient CO2, and B is plants grown in elevated CO2. Now, what's really interesting is in very elevated CO2, 2,000 parts per million, you get a lot of developmental abnormalities. And even though you might see a lot of stomata, we think they're probably non-functional. The stomatal density goes down, but you can see here that you've got two stomata touching each other, and that's on a fully developed leaf in the bottom panel. So a lot of abnormal stomata in elevated CO2. So basically the family of methods we use for the CO2 proxy are, initially they were empirical approaches. We developed training data sets for quantifying the rate of stomatal density response to a known change in CO2 using herbaria or experimental studies. There's also a relationship between atmospheric CO2 and carbon isotopic composition. So both of those methods are empirical. However, there is now a new method, which is a mechanistic-based method. And it basically takes the understanding that assimilation rate and conductance to CO2 is dependent on the CO2 concentration in the atmosphere. And even though we're working with fossils, you can see these fossils are black. So the mesophyll tissue is turned to coal. And lots of experiments have been done to show that the carbon isotopic signature in that coal is pristine, that it's unaltered by fossilization. So we can use carbon isotopic data to estimate internal to external CO2 in that equation. We can count the number of stomata on the leaf surface and look at the size of the stomata to estimate GC total, which is the total conductance to CO2. And then this method can calculate unknown CO2 and unknown assimilation rates simultaneously. So this is basically the basis of the method I use. And then the bottom panel here, this is this leaf surface imaged using epifluorescence, so a UV light source. And even though this is 202 million years old, the cuticle is chemically unaltered. I forgot to include the slide, but if you looked at a FTIR analysis of a modern cuticle and a fossil cuticle, they're almost chemically indistinguishable. They're the same. And you can see here that the stomata and the subsidiaries fluoresce. You can see epidermal outlines. You can see each epidermal cell has a papillae. So we can really observe a lot of the details that we need to calculate this G total in the equation. So the most important equation, I think, for most of my work is this Gmax equation. And this is the theoretical maximum stomatal conductance. So given the number of stomata on a leaf surface and assuming that they're open to the maximum extent, this sets the maximum at which a plant can conduct gases. And we know now by doing a lot of experimental work with porometry and gas exchange that plants typically operate at 25% of their theoretical maximum. So we can calculate functioning of these plants and how they conduct gases in the geological past with this equation and then quarter, so we get the actual rate. But I think the easiest way to explain what I do is actually through cake. So this is a ginkgo leaf surface chocolate cake. It's actually a coffee cake with chocolate icing, where I've reconstructed the epidermal cells with the papillae and the stomata surrounded by fluorescent subsidiaries, and the jelly tots, probably sweets you haven't got, but that's the CO2 molecules going in and the water molecules coming out. So this is my achievement I'm most proud of in my career. I won the Botany Bake Off 2017. So I'm going to jump to our record of how carbon dioxide has changed over geological time. And this is a record over the last 450 million years to the present day. And the CO2 scale is 500 ppm up to 2,500 ppm. And the reason I'm putting it up in this such undigestible form is I wanted to point out that every blue dot on this graph is an individual locality where a team has gone out and dug fossils, collected the fossils, looked at their cuticles, many species usually, many examples, estimated CO2 using different approaches. So they've used the empirical approach, the mechanistic approach, lots of approaches. And then but the blue is actually the average of all of those green dots, and I've put a lowest regression curve through this. And I suppose the first thing you notice is that it's noisy. So it's noisy data, but we can see a long-term pattern. And we can look at this long-term evolution of CO2 in relation to where we're going in the future. So CO2 today is 417 parts per million. And into the future, we're obviously predicting a CO2 atmosphere much more similar to what we've seen in the geological past. And then all of the writing above is all of kind of nearly 50 studies that have generated this CO2 record based on stomata alone. But what we're doing now as a community who work on paleo CO2 is we're trying to improve these records by narrowing the error and also quantifying the error. And these are kind of community-based projects. But what we can see from the record is that at the time of early land mass, 400 million years ago, CO2 concentration was up to 2,000 parts per million. But notice it's a very gappy record. So at the end of the talk, I want to show you some new data. And then we plunged down into the Carboniferous when Indiana was on the tropics, and we had giant lycopsid tropical forests forming coals across the whole Midwest of North America. And CO2 concentrations were similar to today, around 400 to 500 ppm. I'm going to delve into this one in more detail. And then moving into the Mesozoic, the age of dinosaurs, this interval box, I'm going to look into this in a lot more detail. And this is because it was a mass extinction event at the end of the Triassic and the beginning of the Jurassic. And at these events, we can really look at the fossil record and ask, how did a change in CO2 affect vegetation and the functioning of paleoecosystems and plants? And then there's a bumpy ride in CO2 to the present day, but over the last 100 or so million years, when angiosperms, flowering plants, have evolved, CO2 is coming down. So this is my starting point. It's kind of like where we were maybe 5 years ago, and now I want to really focus in on a lot more detail. So what I've been doing with a group of colleagues called the Paleo CO2, it's actually called Paleo CO2 PIP. It's a group of paleo CO2 proxy people. And you can see that there's lots of different proxies. So people who work on boron isotopes of oceans, leaf gas exchange proxy, which is what we call the method we work on, liverwort proxies. And we've been focusing on the last 5 million years because that is the time when our modern flora was evolving. A lot of familiar groups are present on Earth, and it's probably the most relevant in terms of where we're going for the future in terms of climate analogues. And our starting point was a really excellent temperature record, lovely tight errors. This is based on foraminifera. So you can see over the Cenozoic, the last 70 million years, there's been a history of long-term climate cooling. And then we go into the Pleistocene, the last 2 million years, when we have the familiar glacial interglacial cycles. But look at the CO2, it's a horrible mess. So what we did as a community over the last 5 years is really do the hard work where each expert group, the stomatal group, the fossil soil group would work together and make the difficult decision of whether our records, our published records, are good enough. Can we propagate the errors? Do we know the temporal error? If you do temporal error, can you actually quantify the error in an improved way? Do we need to get more data? So we worked in teams during lockdown. That's our kind of COVID project. And we categorized all the CO2 records based on fossil plants, all the different proxies, into 3 categories. And the first category we considered, these are robust records. They're based on multiple estimates. We can quantify the temporal error. We can quantify the methodological error. Category 2 are, I suppose, rejected. So I've got some in here in this reject pile. So they're records in which we recognize we need to do more work to quantify the method error, but we know more that is possible because we have fossil localities that allow us to improve them. And then the last category is, initially it was called the quarantine record. So we didn't like that in COVID. So we made it the archive record. And these are records which we've rejected. The stomatal community have rejected them. The fossil soil community have rejected them because the methods are unquantifiable. They're just not supported by sufficient replication. So based on category 1 record, we've now built a new CO2 record for the last 65 million years of the Cenozoic. So this is the new record, and it's equally horrific in terms of complex data. But the record of CO2 change is in blue, and we're comparing it with a long-term record of temperature change, sea level, and then looking at ice. So some things to point out on this new record. There is a long-term trend of declining CO2 from a peak in the Eocene 50 million years ago. This is when we know the tropics extended up the east coast of North America, where we've got tropical plants right up to 50, 60 degrees north, and extensive tropical forests in South America extending much further than current Amazonian. And then you've got a major transition point between the Eocene and the Oligocene, when we've got a drop in CO2. And then in the middle Miocene climatic optimum, we've got a little bit of an increase up and then decreasing. But some really interesting things emerge from this record. And I think one of the most interesting things is the mismatch in the Oligocene. If you look at the Oligocene, it's a time when Antarctic ice forms for the first time since 300 million years ago. So we've only been in an ice planet since about 30 million years ago. And what's really interesting is that we have a dropping CO2, but a stable, cooler climate. And this is very interesting. It could mean that the temperature properties are wrong here, or it could mean that the CO2 properties are wrong. But what we do have is one interval where there is a mismatch, and the relationship between CO2 as a greenhouse gas and temperature kind of breaks down during that time period. So that's one we're honing in on. But otherwise, there's a very good relationship. During times of high CO2, we see a greenhouse world, global warmth. And during times of low CO2, we see an ice house world, global cold poles, basically. But the relevance of this record for, I hope, this audience of biologists is when you have constraints on how CO2 has changed over the last 65 million years, we can start to look at, what role did CO2 play in the origin of CO2 concentrating physiology, things like C4 photosynthesis or CAM photosynthesis. Probably an even a more graphic way of looking at the record is to use the climate stripes that I'm sure you're familiar with. So each of these colors is dependent on a global temperature change. And you can see there's a really sharp change between the late Eocene, when we go from a greenhouse world, ice-free poles. If we went fossil collecting in Spitsbergen, 87 degrees north, or in the Antarctic, 85 degrees south, we would find fully forested polar latitudes with almost a freshwater cap in the Arctic Ocean with Marsilia water fern in the sea, capped with surrounded by beautiful metasequoia forests and ulmus alder. And then you go into this cooler icehouse climate, which we are in now, where we've got ice caps at the poles. So what we can say from the new CO2 record is that comparing it with the known occurrences and divergences for C4 physiology, is that all known occurrences of crowned C4 lineages, the ancestors of many of the crops that some of you work on in this audience, appear when Cenozoic CO2 concentrations were less than 550 ppm. So low CO2 isn't the only driver of C4 photosynthesis. We know there's many other factors, but certainly we have no evidence of C4 plants in the fossil record when CO2 was higher than 550 ppm. And then for CAM, we have a much poorer record of CAM, and this is because this is crassulation acid metabolism. We cannot use carbon isotopes to detect the presence of CAM, whether a fossil was CAM or not. So here I'm really relying on modern phylogenetic work, which basically puts all the known CAM diversification events when CO2 is less than 420 ppm. So this makes intuitive sense. CAM physiology and C4 physiology are carbon concentrating physiologies, and they evolved in a CO2 world post Oligocene when CO2 was dropped below 550 ppm threshold. Now, what we can also do with this CO2 record is ask the question, which was very contentious among all of the CO2 proxy people. When was the last time current atmospheric CO2 was as high as it is today? And the red line is it was the 2023 CO2 record of 417 ppm. And here's the statistical analysis probability. And you can see that it was the current CO2 as high as it is, the last time as high as it is today, 15 million years ago. 15 million years ago. So in that time, we've had the evolution of grass-dominated ecosystems, steppes in central continental areas. A lot of the grass-dominated ecosystems, I would argue, are low CO2 adapted, relatively, when you look at the long-term history of CO2. And at this point, if we looked in more detail at the temperature, you get a very distinct transition between very stochastic climate, very variable climate, pre 15 million years. Post 15 million years, it's been really quite quiet in terms of climate deviations, comparatively. So now if you look at the last 65 million years, which is on your left, and the future climate projection. So here we have the year 1750 to today and projections into the future. And this is the IPCC data sets. So the last IPCC report. And, you know, without looking at any of the detailed numbers on here, you can see that our future is our geological past. So that's why I would argue, even though the fossil record is biased, it's imperfect, we only preserve a tiny proportion of what was ever living. What we do preserve, we should study and understand, because it's potentially going to tell us a lot about the limits, physiological limits in the future, turnover, resilience, all of these questions that we're asking about global change in the future. So now, for the remainder of the talk, I want to kind of switch gears a bit and now look at a few case studies of fossils, show you some nice pictures of fossils, and look at how we can reconstruct the functional biology of a fossil based on something that's dead. How can we actually bring it to life using modern ecological relationships that have been developed? So I suppose that my group has really been proponents of this, that taking what we understand about how we organize the huge diversity of living plants according to their traits, we want to see, can we take that learning and apply it to fossil plants? And what I'm showing you here is this beautiful, that's a Sphenobiera fossil. It's from the Jurassic of Greenland. And there's a reconstruction. And this is a ginkgo relative that we think could have been a polyploid compared to a bigger genome than modern ginkgo. It's absolutely hugely, it's called Sphenobiera spectabilis. It's literally about this big. And this occurred in the earliest Jurassic after an extinction event. So what I'm going to do now is really step back in time and give you some nice case studies. But first of all, I want to define, well, what is a fossil plant functional traits? So we define it as a measurable property of a plant fossil that is inferred to have influenced the function of that plant when it was alive, and which likely affected its environment or its fitness. So really I want to show you with my case studies is how do we do that inferring? How do we go from an observed anatomy or observed morphology in a fossil to an infer function from fossil plants. So obviously we can't measure photosynthesis of a fossil, but we work with beautiful data sets like people like Scott generate and collaborators. So here's two examples: stomatal conductance and carbon assimilation. So we can't measure either directly, but there's really nice relationships between Gmax, that theoretical maximum stomatal conductance, which is basically a function of how many stomata are there, how big are they, and assimilation rates. And we can see that here for many different evolutionary groups. It's also affected by vein density. So these are low vein density leaves, high vein density leaves. So if you can work out Gmax in the fossil record, which we can, you can infer the functioning, the photosynthetic functioning of that plant's assimilation rate. And then we don't want to just rely on one method, we want to look at multiple methods. So let's say you don't have a fossil leaf, you can observe the stomata, but you have fossil wood or fossil petioles. There's really nice relationships that have been developed between K-leaf, which is the conductance of a leaf or conductance of a stem, and how they relate to photosynthesis. So we can't measure K-leaf as a fossil, but there's a really nice relationship between K-leaf and the distance between a vein and a stomata, and we can't measure that in a fossil. So we go from that distance to K-leaf, which is leaf conductivity, and then we infer photosynthetic biology. And the traits we're particularly interested in. So here's a nice drawing of a fossil medullosa, which was around during the Carboniferous, 300 million years ago, and showing what kind of traits we can take from it. So look, you can look at vein density and stomatal density to estimate paleoassimilation. You can look at leaf thickness and distance between vein and stomata, also to get at assimilation. And then the traits we're particularly interested in are called effect traits. So those are traits which affect an Earth system process. Because what I really want to do is basically improve how fossil plants are represented in paleoclimate models. You probably wouldn't believe it, but a lot of paleoclimate models are run with a modern vegetation. Even for the Devonian, like 400 million years ago, before angiosperms, before grass, they have modern biomes and plants. And that's totally erroneous. So what we want to do is go from the fossil to the traits, to the effect traits, which influence an ecosystem process. And the type of processes we want to measure in the fossil record are those like the hydrological cycle, evapotranspiration, weathering because it's one of the biggest controls on carbon drawdown on a long geological timescale, decomposition, productivity. All of these are Earth system processes, irrespective of the fact that different plants existed in different times. If we can get at Earth system processes, this is what we want to model in a future high CO2 world with a totally different plants, but hopefully the same traits that affect the same processes. So what we've been doing is we have been digging into the modern ecological literature, and there's hundreds of functional traits, but there's a really wonderful book called the Ecological Trait Handbook by Karin, the guy, I can't pronounce his surname, but that team have given you basically a manual of how to measure 30 of the most important contemporary ecological traits that tell you something about how your plant functions today. And we've basically reviewed every single one of these 30 traits and asked, can it be measured in a fossil? If you can measure it in the fossil, did the fossilization process affect the trait value? If it can be measured and we think it is unbiased by fossilization, was it effect traits? Did it influence some Earth system process? So that we've been going systematically through these traits. To give you an example, so I can't believe it's my first fossil slide and it's on many slides in. So here's a lovely array of images of different fossils showing us different traits that you can measure. And it's not so important that you can read these, but I want to just show you the process we followed. So first of all, for every modern trait, we scored it to answer that question, can it be a paleofunctional trait? Can you actually measure it in a fossil? So for example, spinescence, how spiny a plant is, that tells you something about plants herbivore interaction. We can see and measure spines in the fossil record. Or another example would be, also we do that one here. There's palisability. In a fossil, we can observe herbivory and the percent of insect damage, not large herbivores, but insect herbivores we can measure. Cuticle traits like fluorescence, so here are these beautiful glandular trichomes. We can actually measure the pit membrane on a tracheid in a fossil. So that kind of level of detail, if you get the right preservation. Seeds and seed wing will tell you something about dispersal. Distance between rings in wood will tell there's a relationship between lifespan and demography. So you can open the whole concept of demography from an actual fossil observation. But what we did is we scored all those different traits and asked, can you measure it in a fossil? And we got a ranking. And then we said, well, does it affect an Earth system process? So obviously spinescence does not affect an Earth system process. So although it's measurable, it was downgraded because it's not going to tell us about hydrology. It's not going to tell us about carbon. And then we asked, what went one further step, we asked, is this plant functional trait or paleofunctional trait currently, can it be parameterized or is it parameterized in a paleoclimate model or a paleo-ecosystem model? And based on all of those scoring criteria, we came up with a ranked list of paleofunctional traits. And these are our traits that we want to focus on, and then we've kind of guided the community as targets for further study. And just to illustrate, the ones that came out, almost not surprising, photosynthetic race, yes, it's very important. We want lots of tools to measure this in the fossil record. Maximum stomatal conductance. Another important one here is leaf mass per area. And this is an ecological race within the leaf economic spectrum, which I'll talk about a little bit further. So now I'm going to give you examples of how we apply this method to different intervals through Earth history. And we're going to start 110 million years ago. This is the Middle Albion, and it's a locality we collected in West Greenland, just above Disco Island. A lot of years ago, I collected this. So to place you in the evolution of plants, the arrow indicates the time I'm looking at. It's basically a transition point between the mesophysic, the middle plant world, gymnosperm, conifer-dominated world, and cenophysic, young plant world. So this is angiosperms. So angiosperms are present. We know they're present on the landscape, but they're not ecologically dominant. And in terms of global biomes, climates, it's an ice-free world in the Middle Cretaceous. Here. So it's an ice-free world, and actually you'll see very little tropical forest. It's pretty hot and dry world. So the localities occur in West Greenland. It's an absolutely beautiful part of the world. We camped on the shores. Our breakfast views every morning were fin whales and pods of whales. You can literally hear them before you see them. There was no water, so we actually had to pull little mini icebergs out of the sea and then melt them in a tarpaulin as our water source. Absolutely wonderful field work. And we were looking for the earliest angiosperms in the early Cretaceous in the Arctic. They're already present in the tropics, but we're looking for them. And not just looking for them, we're trying to see functionally, how did they compare with their ancestors, the angiosperms? So we found a lot of non-angiosperm fossils. So we found, and typically in paleobotany, you use a morphological species concept. So you morphotype the plants. So we find things related to Cyanopiscus, conifer that occurs today in Greenland, in Japan. And it's got a lovely suture down the middle where all the stomata occur. So that's definitely Cyanopiscus. and two other types of conifers that are extinct, Caryophyllaceae and Moraviaceae. And these are cuticles, so they're 110 million year old cuticles, and they're fluorescing under UV light. We did find rare angiosperm cuticles in these rocks. And if you look at this piece here, your eye is kind of guided to these bright glowing spots on the leaf surface, and I think these are probably pellucid dots. These are glandular cones, and you have to kind of focus in to see the actual stomata on these surfaces. So there's a nice stomata in relation to the gland, and there's a cross section of the trichome there. So you can see the preservation is amazing. So we applied a functional trait approach, and just as an example, we compared what was the photosynthetic, actually, what is the gas exchange rates of these early angiosperms in the Arctic compared to some of the conifers that they were co-occurring with. And I'd say the forests were wet and warm. So Pseudopiscis today occurs in 3 meters of rain, constant humidity, annual rainfall of 3 meters, 3,000 millimeters. So in green is our angiosperm. This is the log the sediments from which we collected the fossils. They're around 110 million years old. And you can see here that angiosperms were functioning very similar to their gymnosperm ancestors. They had very similar conductances. They weren't this supercharged, fast photosynthetic rate, high conductivity compared to their ancestors yet. But as CO2 starts to drop, this is when angiosperms really change their morphology, their vein density, their stomatal density, and they change their photosynthetic biology and outcompete these groups, the red and the blue, but it's not shown here. But really, this is just an illustration of how you can use this functional trace approach on fossils. Now we're going to jump back to the Triassic-Jurassic boundary. And I've done a lot of work here, a bit of an obsession with Greenland. So now this is East Greenland. And this was a time of mass extinction in the fossil record. So we're firmly in the Mesophytic. So it's gymnosperm dominated. We have lots of cycads, very diverse ginkgos, lots of Araucaria conifers, ferns, a group of phenatites, which are an extinct sea plant group. And we were looking at how the vegetation responded between the Triassic and Jurassic. And we showed, although I'm not going to show it today, we showed an 85% extinction of species under a global warming. So a CO2 change between about 1,000 ppm CO2 and 2,000 ppm CO2. But what we were particularly interested in is how did those forests pre and post the extinction event, how did they function? A different composition of species can function the exact same way, or it can function differently. So we're particularly interested in the functioning. So we used the traits we were interested in looking at was leaf mass per area. And it was Ian Wright in Australia and a huge group of collaborators who came up with this concept of the leaf economic spectrum. And basically every single species on the planet can be placed in terms of its leaf traits on this spectrum of photosynthetic rate, leaf mass per area, and other traits, so nitrogen content. And there's a spectrum or a continuum of traits. So plants live fast, die young, high nitrogen carbon, and usually a low leaf mass per area. So that would be your dandelion, or I suppose an oak leaf, six months, a deciduous oak. On the other end of the spectrum, you've got leaves with a long leaf lifespan, and the extreme would be 27 years, or a monkey puzzle tree. 27 years, it's huge investment in structural compounds. It has to survive a long time. It's got low nitrogen, low photosynthetic rates comparatively, and a high leaf mass per area. So we were trying to, we knew that if you can develop a proxy for leaf mass per area, it would allow you to get onto the leaf economic spectrum and place an extinct fossil into the spectrum of function. which is known by modern ecologists. So if you look at this beautiful fossil down here, this is from Greenland, Sphenobiera. And in a fossil, we can observe size, but we can't measure mass. We can't measure density. So we developed a relationship between cuticle thickness, which is a very dense tissue, and leaf mass per area for gymnosperms. And you can see there's a lovely log-log relationship between, with increasing cuticle thickness, you get increasing leaf mass per area. And this is a cross-section of a ginkgo. You can see the upper cuticle, and it's, you know, over 200 million years old, and the lower cuticle, we can check its cuticle by looking using UV lights, and it autofluoresces. Initially, I was really excited because the internal tissue I thought was mesophyll. It really looks like mesophyll, doesn't it? But I think it's an artifact. I think it's coal that is fractured in a very mesophyll looking shape. So I think we need to do more work on that, but you know, we've rejected the idea that it is mesophyll. But the important thing is that you can measure cuticle thickness across the length of a leaf. And with those measurements, you can estimate leaf mass per area and place this extinct plant into a modern ecology, modern functioning space. Now, this is a really horrible graph as well, but what we did is, I'm going to walk you through it. This is the extinction event. And in the peak of the extinction, we lose nearly all trees in Greenland. So you just get a fern-dominated landscape. The trees disappear, they migrate somewhere. They don't go globally extinct, they go locally extinct, but they come back. We reconstructed CO2. You've got CO2 going from 1,000 ppm to over 2,000 ppm and back within a space of maybe a few million years. And we have a relative abundance data for all the taxa. So we know ecologically which are dominant, which are rare, and how they're changing. So we measured leaf mass per area for Ginkgo ales and Bennettitales, the dominant plant groups. And then we worked out a community-weighted leaf mass per area. And that's what this is here. So what the data show is that the forests of the late Triassic, before natural global warming occurred caused by volcanism, the plants had a very deciduous ecology, similar to modern ginkgo. The high CO2, there's a strong selection for plants with a high leaf mass per area, basically a tolerator strategy. And you see extinction of plant groups of fast assimilation rates that have this kind of live fast, die young, high nitrogen, fast enzymatic rates, and a selection for things that are slower. And the forests of the Jurassic have an evergreen ecology. So it seems that there is strong selection during a global warming event for an evergreen ecology, and that we see this on geological timescales. And we've actually seen this when you grow these plants in a super elevated CO2, 2,000 ppm, they greatly extend the leaf lifespan. So ginkgo, which usually drops its leaves, you know, after 3 months, it's almost becoming evergreen. It's hanging onto the leaves. It's got really high leaf mass per area under high CO2. And I think this is very interesting as a kind of a hypothesis to look at for the future. You know, how are crops, how are maybe not so much crops, but some of our forests, will there be a strong selection for this evergreen evergreenness in a high CO2 world in the future? Fast one going. Okay. So the next step is really deep in time. So 300 million years ago. And this is the late Pennsylvanian or Carboniferous, the age of carbon. And we're going to look at North America. So imagine Indiana, Illinois, as coal swamps 300 million years ago on the equator. So now we're in the Paleophysic. In terms of the overall ecology and evolutionary biology, although seeds are present, the earliest seeds had evolved in the late Devonian, the world is still ecologically dominated by spore-bearing plants. So giant lycopsids, Pecopterus ferns, a lot of those wonderful fossils are preserved as coal balls, which are sitting in a huge big warehouse in Tom Phillips in Urbana-Champaign. So again, we reconstructed CO2 records through this time interval, the late Pennsylvanian, so from 311 to about 290 million years ago. And this CO2 record is a consensus record based on the CO2 proxy and paleosol proxies. And what was really exciting about this record, and kind of somewhat expected, is that you can see glacial, interglacial transitions in the Carboniferous. And the Carboniferous had always been a very contentious time because climate modelers, so you've got glacial, interglacial transitions in the Carboniferous. So this was the last time the Earth was in an icehouse state before a current icehouse. The lower bound, the CO2 at the low point in the glacials, is almost identical to that of the Quaternary. 190 parts per million is the lower bound. The Earth's CO2 has never gone below that because we've got feedbacks that kick in that stop us going into an icehouse planet. But interestingly, during the interglacials, the warmer phases, CO2 concentration is much higher than the Quaternary. So it's about 600 ppm. So the swings are bigger. And then the periodicity here is 400,000 years. And that's a Milankovitch periodicity. So that was extremely exciting to see that. But once you've got the context of what is the changes in CO2, you can begin to look at the fossils and look at their functioning. So we looked at fossils. If you go to the Field Museum, you can walk through that lovely diorama. You've got the giant lycopsids, Calamices, Equisetum relatives, Isoetes relative, and this is an extinct seed plant group called Medulosa. These are leaves of those extinct seed plant group. They're called Allothopterus. So it looks like a fern, but it produces a seed on the adaxial surface. So a totally extinct group. And this is Neuropterus. These are glands which your eyes are probably drawn to, and you have to look down to see the stomata. But we reconstructed the GMAX, the diffusive biology of these plants. And it was a huge surprise. These two extinct groups have stomatal conductances, which are nearly double modern angiosperms. And really, Paleobotans were very hesitant to call these tropical rainforests because they're not angiosperms. These are spore-bearing plant groups and extinct seed plant groups. But functionally, they were functioning like tropical angiosperms. These ones up here. So if you measured tropical modern angiosperms, and that's based on looking at their stomatal biology. Now, we've learned since that they've probably come down a little because they're constrained by their stem conductivity. But that was a big surprise because it was always argued that angiosperms, this new group, this new dynamic evolutionary group that we, all our food is based on and the whole world is powered by currently, were new and shiny and more functional, faster. But actually, in actual fact, in the Carboniferous, in the icehouse, we have forests which were functioning very similarly to angiosperms today. And we've tried to estimate the photosynthetic biology of these plants using lots of the proxies and also modeled it. So we've modeled it for modern day ambient oxygen, 21%, and 35% oxygen, because this is probably the only time in Earth history when the amount of carbon being sequestered and buried in swamps massively overwhelms the amount of carbon being released by degradation. So the net effect of that was that atmospheric oxygen increased over this whole interval. The byproduct of our coal today was a high oxygen in the Carboniferous. And I suppose what I wanted to point out from this graph is that our modeled assimilation rates based on a paleo biome model. And our estimates based on observing vein density and stomatal density and estimated assimilation rate, they're very well aligned. So that kind of gives us a lot of confidence that ultimately we can actually build a record of how photosynthetic biology changed through Earth history based on fossil observations, not models. There's lots of models. We want to actually observe it and reconstruct it. And then if you've got a functional trait of a fossil, you can actually use that functional trait to try and classify the biome in which the fossil occurs. So these are water use efficiency estimates for an interglacial in the Carboniferous, and they're the water use efficiency values for the plants. The plants aren't so important. Now, this is a modern data set, and it's a measure of water use efficiency based on stable carbon isotopes. For tropical forests, for the 1980s and today, for tropical seasonal forests, moist, so this is, that's Fiji, these are forests in Puerto Rico, moist and dry, and subtropical deserts. And if you look at the water use efficiency values, they are really low. So these plants, they were living in swamps, they had really low water use efficiency. They were basically like open pipes, and they were functioning like modern day tropical forest biome. And what's interesting is on geological timescales, we see an increase in water use efficiency with increasing CO2. And we see the exact same pattern on historical timescales. Modern tropical forests increase their water use efficiency with increasing CO2, as you'd expect. There's more CO2 in the atmosphere, the plants develop fewer stomata, they get the same carbon gain, but they reduce their water loss. It's classic, optimization, optimization behavior. So here's a really nice example of evolutionary responses of plants are mirrored by ecological timescale responses we see in modern ecosystems. Ultimately, what we want to do is take those traits. So here we have water use efficiencies for these two different groups, the lycopsids, which made up the coal swamps, they were like open pipes, and the medulosa, which were more water-wise. So the lycopsids are here, really low water use efficiency, and the medulosa is higher water use efficiency. So what happens to an Earth system process when you swap a lycopsid-dominated ecosystem? So a swamp full of these giant lycopsids with a swamp full of medulosa. And it has an effect on runoff. It has an effect on the hydrological cycle. And we can see it here. So this is a paleo ecosystem model. And you can see that the amount of runoff is low when you've got lycopsids, and it increases hugely when you swap the ecology to medulosas. And this is because when it rains, if you have high conductance, you transpire a lot and you recirculate the rain. If you have a lower transpiration, instead of the rain, I'm confused, instead of recirculating the water when it rains, you balance that sun by increasing runoff. So a prediction in a future high CO2 world is that we will see more runoff, more flood risk. And we can actually test this in a high greenhouse world of the past. And with increased runoff, you'd expect to see floodwaters going into the ocean and lots of sediments in the ocean. We see examples of this. With the change from these plants to these plants, you see pulses of debris in the marine fossil record at the right time, which supports the idea that you're changing this Earth system process, changing runoff because of a simple change in the traits of the plants. So this is about my last slide now. The whole aim is to use observational data on fossils to infer functioning, and then use that functional biology to better represent fossil plants in a paleo ecosystem model, to try and understand how did plants change the world? How did they terraform the world? How did they change weathering? How did they change hydrology? And can we take any of those inferences from the past and apply them to the future? Oh, I forgot I'd added one piece. Has everybody had enough advance now? Because I can skip to the final. I think I am going to have the I'm going to skip that one. It's too much. What I will just tell you one thing. Okay, so the last one is the Devonian, and I've been looking at fossils from Ireland, a really famous fossils called Archaeopteris hibernica. And these were trees. This is about 2-meter diameter, huge trees, which we estimate had carbon sequestration potential similar to modern kauri pine, absolutely massive. But there were some of the earliest fossils with leaves in the fossil record. So the earliest example of a fossil with leaves, up to that point, that all plants were leafless. And here are some of the examples of the leaves. So they look like ferns, but they're actually attached to gymnosperm wood. So it's a hybrid, which is now extinct. Fern-like leaves, conifer-like wood. And nobody had kind of understood the leaves. So we thought, okay, let's try and get the leaf mass per area for these plants. And in orange is the leaf mass per area. So the fossils, which are just beautiful, but no one understood their biology. It looks like they're really thick, really dense leaves that have a higher leaf mass than any of the living gymnosperms. So even though they look so delicate as fossils, everybody presumed they were kind of fern-like, very delicate. It didn't make sense, but it's just a ghost of the fossilization. These things were probably extreme evergreens with, leaf lifespans, maybe 30 years, some of the earliest leaves. So to conclude, advances in CO2 property research and a community approach are continuing to improve our understanding of atmospheric change through Earth history. And a paleotrade approach, we think, shows promise to elucidate the evolution of species and ecosystem function and test the influence of plant trait evolution on Earth system processes. So thank you very much, everyone.
We have a room for a while, so we've got time for questions.
Yeah, I'm happy to take any questions.
I'm struggling to put this in question form, so maybe like an example would be better. But say like in a million years, someone's looking at fossils from our plants and see a gymnosperm with very leaves. What's to stop them from assuming there must be dinosaur herbivory happening today based on that? Like, how do you assume the fossil traits that you're looking at are not just like a carryover or like are adaptive to the environment?
Okay, I'm with you. So that it's phylogenetic rather than adaptive to the environment.
Mm-hmm.
Good question. I think because experimentally we've looked at many different groups, so which are good analogs, and within an 8-week experiment, across the board, we see like a really strong increase in leaf mass per area in Dixonia, Cyathea, Araucaria, Agathis, Nagia, all of these different plant groups. There's never universal in biology, but leaf mass per area is one of those that really changes. So, and that's across many different phylogenetic groups. What you could do is you could obviously use statistics which take into account phylogeny. We haven't done that yet. I mean, really, we're just kind of testing what traits are the best to focus on, what traits are going to be meaningful to get it functioning. And to be honest, what we want to do is reconstruct the functioning of the whole ecosystem. I don't really mind if it's a response to an environment or it's a feature of evolution. I just want for that time, that place, how did it influence the hydrological cycle? How did it influence bioecology? That's ultimately what we're trying to do, better parameterize the actual Earth system models. Yeah.
How sure are they that those lepidolite forests were flooded with water quite high up to the top of the canopy?
Oh, to the top of the canopy.
Because like the stem is now constraining photosynthetic rays,
Yeah.
but that you were saying that the gas exchange estimates are going to drop because of the stem, which is rare, which is very uncommon for the stem to economic gas exchange. It's mainly in the leaf.
Yeah.
So, but it wouldn't be if the stem was in water.
Oh,
Do they know?
No.
What's the record whether they were in water or not?
they were in peat swamps.
Yeah.
They were in water for definite, and they were, you know,
Or how deep it was. Yeah. There was no runoff. Was it rain? Was it more like that?
No, so the sedimentology tells us it was a peat.
Yeah.
It could have been a fen, you know, they can't tell between a fen and a peat, but there's a lot of cobalt, which are carbonate concretions, which would suggest it could be a fen, spring water fen. Oh, I don't know whether they know that, but it's interesting. There's a guy, Walton Green, he's suggested that these giant lycopsids could have had a carbon concentrating
Yeah.
biology, where they were taking root-respired carbon that was coming up the xylem stream and capturing, so not CAM, not C4, but some sort of CO2 concentrating. But it hasn't really been tested from lots of different angles, but I think it's a very plausible hypothesis. Well, I don't know. I'll have to come back to you. I'll have to ask a sedimentologist that.
Yeah.
You know, maybe they can tell how much standing water there was. These could have been, you know, if it's like modern tropics, they could be seasonally flooded very high.
Yeah.
The lycopsids were in the peat, but some of the other groups were on supposed terra firma.
Yeah.
And they were on the margins of rivers and lakes. Now they could very well, and a lot of the fossils are preserved in floods. They're called crevice splay, so it's a flood water sedimentation. So it could be those ones could have been flooded high.
Yeah.
Yeah. So why is it that they? So what you say, the stem does limit transpiration if they're in high flood water?
Well, no, it never seems to, but if those gas exchange numbers are getting reduced because of the stem, it seems very unlikely. That seems like more of a leap of faith to believe that the stem would ever limit transpiration in the leaf.
Yeah.
Because you would easily overbuild the xylem. You can easily overbuild hydraulically oversupply with a stem.
Now, also, you have to remember that the oxygen concentration is higher, so the density of the atmosphere is different, and it affects CO2 and oats, water diffusion differentially.
Yeah.
Yeah.
Well,
Can I ask an odd question?
Please. They're always the best ones.
Why are there no herbaceous gymnosperms, or were there in the past?
Very nice question. So of course, we've got no living herbaceous gymnosperm, and there is one in the fossil record, putative herbaceous gymnosperm, that was published by Gar Rothwell. And herbaceous conifer. So any other herbaceous gymnosperm in the fossil? No, it was just one, Gar Rothwell. So it's really interesting. Yeah.
Why?
I don't know, why not? So some people actually say that the angiosperm, there's so many innovations in angiosperms. You know, double fertilization. Okay, Nitates do that as well, but the flower, bisexuality, but herbaceousness is an angiosperm. Okay, ferns as well, but compared in seed plants, herbaceousness is fairly dominantly an angiosperm trait that we just don't see in the gymnosperms. Ginkgo, Nitates, conifers, don't see it. None of the extinct groups. Yeah, so it is really interesting. Yeah, there's just one, but a lot of what we do, I suppose, is trying to work out. You have a leaf fossil, and you're trying to work out demography. So you can do it from a leaf, but you could do it from wood, but that's not going to help you with herbate, So you need a chance fossilization event of an entire plant, and that's really rare. Yeah.
These are long-term changes that come millions of years.
Yeah.
What happened rate of change? It can plot any rate of change?
The rate of change of what? CO2?
Any of the
Okay. A function. Oh yeah, good question. Well, so in the most extreme global warming events in the geological past. So they're called hypothermals, like the Triassic-Jurassic boundary, the Paleocene, Eocene. The rate of change is about 2 orders of magnitude slower than today. Much slower. There's no comparison to today.
Can we predict the effect of these changes on our food security?
Based on the past,
That's what we should have, we won't have to.
on food security, no, I think what from the paleobotany, we know that the earth is fine, the biosphere is fine. There will always be green, we will not have a dead world. We can go to 2000 ppm and not have a dead world, but the ecological composition will be completely different. Completely, and natural, and things like C4 biology. You know, we're reaching that limit now, C4 biology in a natural system where it's competing with the C3 plant. And the rate of change, the plants have always been able to respond if the rate of change is sufficient to allow them to migrate somewhere and then come back. And that's what they tend to do in the fossil record, not adapt in place. They move where they would end up come back.
I've seen other mutation rates, which is connected.
Okay, yeah, mutation rates. I think that the only way you can do that is with modern phylogenetics and with a good time tree. So that's where fossils actually give you a age constraint. Yeah, yeah.
Sure. Right here, right now. Yeah.
Oh.